This is our introduction into the muscle unit. We will be talking about muscle functions, muscle tissue types. We'll be talking about how muscles are structured. We'll be talking about, of course, some muscle terminology that will help us move forward in our muscle unit. So let's begin with muscle functions. All muscles produce movement. That's their big function, is to produce movement. The other three here, maintaining posture, stabilizing joints, generating heat, all of these are really the province of skeletal muscles, which is, of course, where we'll be focusing on with this unit. The different things about muscle tissue, these are elongated cells, and they are contractile, which means they're built to change shape, to shorten and become thicker. And what they do is they pull at their attached ends, thickening in the middle so that they can create movement. There are three types of muscle tissue. There is skeletal, smooth, and cardiac. And this chart is really about distinguishing between those three types of muscle tissue. Skeletal is the one we'll be focusing on. Cardiac we'll touch on in our cardiovascular unit. This is really only found in the heart. And then you have smooth muscle. This is found in the digestive system and in your blood vessels. And we'll be covering most of the smooth muscle tissue in your body. We'll be covering in our digestive unit. It's always good to take a look at this chart and really look at the differences between the three types of muscle tissue, just so you can be clear and know what you're looking at when you look at a skeletal muscle. So let's focus on skeletal muscle functions. It is all about producing the movements for your skeleton. So really moving those bones around. Of course, these muscles are attached to your bones. And the biggest distinguishing factor or really distinguishing feature of skeletal muscle is that it's striated, which means if we look at this picture, you can see some skeletal muscle and you can see those striations. Striation means that it's striped, and you can definitely see all of those really unique striping appearance here. This is just another picture to kind of show you that striated feature. This is a cross-section of a muscle, and what you see here is the biggest thing about muscles, which is they are bundles packaged together into larger bundles that are packaged all together with other bundles into a much larger package. So really, muscles are just bundles within bundles within bundles. And we're going to go through this picture to really talk about what each of those bundles are. You first have the largest bundle, which is, of course, the muscle, and it is covered in what is called the epimysium. That is the packaging, kind of like gift wrap, that just surrounds the muscle, keeping all of those bundles inside together. You can see, of course, we have blood vessels supplying our muscle. They're all throughout this structure. Then we have these larger bundles here. Those are called fascicles, and they are wrapped in their own kind of gift wrap, which is called perimysium. Make sure that you know perimysium and epimysium, and you know the difference. That will be on your test. Then, within our fascicles, which you will need to know, you have muscle fibers, and these are actually the cells. These are muscle cells, and they are really interesting, and on the next slide, I'll really break them down. So make sure that when you are looking at muscle structure, that you understand that you start out with muscles, and they are just a whole bunch of fascicles bundled together, and a fascicle is a whole bunch of muscle fibers bundled together. Make sure you know that order of how a muscle is arranged through its anatomy. That will be on your test. So let's look at a muscle fiber deep down in here. And when you look at a muscle fiber, what you notice is that it is actually a whole bunch of myofibrils packaged together. And they are actually surrounded by their own membrane. That is the sarcolemma. That is what we call the membrane on a muscle cell. So the sarcolemma here. Okay? And so you have this muscle cell, and it's full of these myofibrils. And myofibrils are packed full of nuclei, and they also have these contractile units. These are the things that are actually producing movement when you move a muscle. These are the guys that are actually producing that movement. There are two shapes I want you to be aware of here. If we look down here, you can see the thin line and you can see the thick line. The thin line is the actin filament and the thick line is the myosin filament. And I want you to remember those because we're going to be talking about them in just a little bit. The reason that those are so important is because they are the things that are actually producing movement. They're the contractile bits. The sliding filament theory is the explanation for how muscles contract. We used to think that it was just like a coil, like a big slinky, and that slinky just contracts and reduces its length. We thought that that's how muscles worked, and we didn't understand how it all worked. But with the sliding filament theory, we actually understand that it's not like a slinky at all. It's actually like two platforms sliding across each other. And those two platforms are, of course, our actin and myosin fibers. And they slide across each other, creating cross bridges. And of course, they're using up ATP. And I'm going to get a little bit more into how this happens on the next slide, where I have some pictures to kind of demonstrate how that works. So I want to draw your attention down to this picture right here. This is a close-up of actin and myosin filaments. The pink one right here, where my cursor is moving, is of course actin. It's the thin filament. And these pink beads that you see, those are the actual actin proteins. And they're being guarded by some other accessory proteins. Then down here we have the myosin filament, and they have these lovely myosin heads that really want to attach to actin. And these protective proteins right here, the troponin 1 and troponin C, protect the actin from constantly being attached by the myosin. So what they do is kind of act as a guardian and kind of prevent the myosin from binding to the actin all the time, because these two proteins are constantly attracted to each other. So when it is time for a muscle to contract, we'll get a nerve impulse, and I'll go a little bit more into nerve impulses down the line. But when we get a nerve impulse, that is going to cause a whole bunch of calcium ions to flood into our myofibrils, and those calcium ions attach to these protective proteins on the actin filaments. And they cause them to go away or to move out of the way so that myosin can actually bond. And myosin kind of latches onto actin and kind of pulls it. And you can see the arrangement of how actin looks. So here you have your feet from your myosin little proteins. And here you have your actin filament. And that myosin is going to attach right there and then pull it towards the center. So each of these is going to move inward like this. And as you can tell, that produces a contracted shape. That's where we're getting that thickening happening. And the amazing thing is that you have thousands of these in every single muscle, and these are the guys that are actually producing movement. This is the only thing that muscles can do. All they can do is contract and release. So let's talk a little bit about the releasing mechanism here. What happens is ATP attaches to the myosin and actually causes it to release from actin. This is why you get cramps when you run out of oxygen, because you don't have enough ATP to stop that contracting. I've run out of time on this video, And